Welcome, curious minds, to another deep dive. Today, we're really getting into it, exploring the very blueprint of life itself.
That's right, we're digging into a fantastic source, Karp's Cell and Molecular Biology, 8th edition, specifically Chapter 10, The Nature of the Gene and the Genome, foundational stuff.
Absolutely. Our mission today is, well, to sort of distill centuries of discovery, from the earliest ideas about inheritance right up to modern genome sequencing. We want to make it clear how genetic information is stored, how it's passed on, how it's expressed, hopefully with some real aha moments for you.
It really is an incredible story, just watching how the idea of a gene has changed over time, from these sort of abstract concepts to the actual physical DNA molecule and then the whole genome. It's quite a journey.
Okay, so let's unpack that journey. Where do we begin? Mendel, right, back in the 1860s.
Yeah, exactly. The 1860s, long before anyone really knew what a gene physically was.
In that garden, in the Czech Republic now.
That's the one. Gregor Mendel, an Austrian friar, doing these incredibly careful experiments with pea plants.
It sounds so simple, crossing pea plants.
Oh, yes and no. His genius was in the plan. He chose traits with clear alternatives, tall or dwarf, yellow or green seeds, things like that. And then he just counted, meticulously, generation after generation.
And from just counting peas, he came up with these huge ideas, still fundamental today.
Absolutely. He figured characteristics must be controlled by distinct factors, what we now call genes.
And that you get two copies, one from each parent, the different versions being alleles.
Exactly.
Yeah.
And that one allele could mask the other dominance.
Right.
Plus, that these factors, these alleles, they separate when making reproductive cells. Gametes. So it's a gamete only has one copy. That's segregation.
And independent assortment, too, trait sorting independently.
Well, we'll get back to that one.
Yeah.
But yeah, segregation and assortment, basic laws. But here's the kicker. His work, published in 1866, was basically ignored for 35 years.
Wow. Really?
Yeah. Rediscovered independently around 1900. He was just way ahead of his time.
So, okay, Mendel figures out these factors exist, but the next question is, where are they? Inside the cell, where?
Right. That question started to get traction later in the 19th century, looking at fertilization. Scientists like Theodore Boveri, working with sea urchin eggs, he noticed something odd.
What was that?
If an egg got fertilized by two sperm polyspermy, the embryos went haywire. Cell divisions were chaotic, they died early. So Boveri concluded that normal development needs a specific combination of chromosomes, suggesting they weren't all the same, they carried different information.
And wasn't there work on roundworms around then, too?
Yes, Ascaris. Eduard van Beneden saw that body cells had, say, 4 chromosomes, but the sperm and egg nuclei before they fused only had 2 each, half the number.
Which lined up with Weismann's theoretical idea of a reduction division.
Exactly. August Weismann proposed meiosis must involve having the chromosome number in gametes. Otherwise, it would double every generation, right?
Makes sense.
Then, the big connection came in 1903. Walter Sutton. He was looking at grasshopper sperm cells.
And what did he see?
He saw the chromosomes came in pairs that looked alike. He called them homologous chromosomes.
Like Mendel's pairs of factors.
Precisely. And crucially, during meiosis, he watched these homologous pairs find each other, form these structures called bivalents, and then separate into different daughter cells.
That's it. The physical basis for Mendel's segregation. You could literally see it happening.
You could see it. It was the bridge between abstract genetics and concrete cell biology.
But hang on. If genes are physically on these chromosomes, wouldn't all the genes on one chromosome stick together? Linked?
Yes. The linkage problem.
That seems to contradict Mendel's independent assortments, where different traits seem to shuffle randomly.
That was a huge puzzle. And the answer turned out to be, well, Mendel got a bit lucky.
Lucky how?
The P traits he studied happened to be on different chromosomes, or so far apart on the same chromosome that they looked independent because of crossing over. But Sutton actually predicted this. He proposed linkage groups. Genes on the same chromosome should be inherited together, and pretty soon, researchers found examples, like in sweet peas.
Which leads us, I guess, to the fruit fly, the workhorse of early genetics.
Exactly. Drosophila melanogaster. Thomas Hunt Morgan's lab at Columbia University, starting around 1909.
Why fruit flies?
They were perfect. Short generation time, like 10 days. Loads of offspring, small, easy to keep in the lab. Morgan's team had to find their own variations, though. The first big one was a white-eyed male fly, a mutant.
And that white-eyed fly was key, wasn't it?
Oh, definitely. Because they quickly figured out that eye color was linked to the sex chromosomes. More proof genes were on chromosomes.
Okay, so linkage is real, but then things got even more complicated, right? The linkage wasn't perfect.
Exactly. Genes on the same chromosome sometimes didn't stick together. They could get reshuffled. There were unexpected combinations in the offspring. Recombinants, crossing over. Genetic recombination. Back in 1909, a Belgian scientist, Janssens, had actually observed homologous chromosomes physically wrapping around each other during meiosis.
Forming those X shapes. Chiasmata.
That's them, chiasmata. He suggested they might be points where chromosome segments break and exchange.
And Morgan put that together with the recombination data.
He did. He proposed that this physical crossing over was the reason for the genetic reshuffling, for the recombinants.
And then came the really big leap from one of Morgan's students.
Alfred Sturtevant, an undergraduate. In 1911, he had this brilliant insight.
Which was?
He realized the frequency of recombination between two linked genes seemed to be constant. And he hypothesized that this frequency was proportional to the physical distance between them on the chromosome.
Wait, so the further apart two genes are?
The more likely a crossover event will happen somewhere between them, separating them.
So you could use the recombination frequency to map genes?
Exactly. To figure out their relative positions along the chromosome, Sturtevant started making the first genetic maps for Drosophila, a revolutionary idea. Map the invisible.
That's incredible. So we knew genes existed, we knew they were on chromosomes, we could even map their locations, but what were they, chemically?
That remained the big mystery for decades. What is the actual stuff of heredity? The focus really shifted in the 1940s.
The story starts earlier, with finding DNA.
Much earlier, 1871. Friedrich Miescher isolates this stuff from the nuclei of white blood cells, actually from pus on surgical bandages.
Charming.
He called it nuclein, acidic, rich in phosphorus. And remarkably, someone named Otto Hertwig, way back in 1884, speculated this nuclein might be responsible for heredity.
But that idea didn't stick.
Ironically, no. As scientists learn more about its building blocks, thanks partly to Phoebus Levine.
He figured out the deoxyribose sugar, right?
He did. Crucial work. But he also proposed the tetranucleotide theory. The idea that DNA was just a simple, boring repeat of the four bases A, G, C, T, A, G, C, T over and over.
So it seemed too simple. Not enough complexity to hold genetic information.
Exactly. Proteins with their 20 different amino acid building blocks seemed way more complex, much better candidates for carrying the genetic code. DNA was sort of dismissed.
Until some key experiments turned things around. Griffith.
Frederick Griffith, 1923. Working with Streptococcus pneumonia, the pneumonia bacteria, he had two strains, a virulent S-type with a smooth coat and a non-virulent R-type that was rough.
Okay.
He found if he injected mice with heat-killed S bacteria, nothing happened. If he injected live R bacteria, nothing happened. But if he injected heat-killed S together with live R,
the mice got sick.
They got sick and died. And he found live S bacteria in them. The harmless R cells had somehow been transformed into the deadly S-type by something from the dead S cells.
The transforming principle, but what was it?
That's what Oswald Avery, Colin MacLeod, and Maclean McCarty set out to discover at the Rockefeller Institute. Took them a decade of really painstaking work.
And they nailed it down.
They did. In their 1944 paper, written very cautiously, they presented strong evidence. The transforming principle had the chemical properties of DNA. No protein contamination was found. And crucially, only enzymes that specifically degraded DNA destroy the transforming activity. Enzymes that broke down proteins or RNA had no effect.
So it had to be DNA.
It really pointed strongly to DNA. But the scientific community was slow to accept it. Protein was still the favorite. McCarty later recalled speaking about it to maybe only 35 people left in the audience.
Wow. But Avery knew how big it was.
Oh, yes. In a private letter, he wrote, "If we are right, then it means that nucleic acids are not merely structurally important, but functionally active. This is something that has long been the dream of geneticists." He saw the implications.
And then Hershey and Chase sealed the deal with bacteriophages?
Pretty much. Their 1952 experiments, using viruses that infect bacteria, showed elegantly that it was the viral DNA, not the protein coat, that entered the bacteria and directed reproduction. That, plus Chargaff's findings, really shifted the consensus.
Yes, Chargaff. What did he find?
Erwin Chargaff, around 1950, was analyzing the base composition of DNA from different species. He demolished the tetranucleotide theory by showing the amounts of the four bases A, G, C, and T varied widely between species. DNA wasn't simple repeats.
So it had the necessary variety.
It did. And crucially, he found fixed ratios within a species. The amount of adenine, A, always equal the amount of thymine, T, and guanine, G, always equals cytosine, C, ATGC. Chargas rules.
Which didn't make sense structurally yet.
Not yet. But all these pieces, DNA is the transforming principle, the Hershey-Chase result, Chargas rules were on the table. The stage was set.
For Watson and Crick, 1953, the double helix.
The iconic moment.
Yeah.
James Watson and Francis Crick, using X-ray diffraction data crucially generated by Rosalind Franklin and Maurice Wilkins.
Yeah.
They built their model.
And what were the key features of that model?
Okay, several things. One, two chains, two strands of nucleotides, wound around each other in a right-handed double helix. Two, the strands run in opposite directions. They're antiparallel. Three, the sugar phosphate backbones are on the outside, making the molecule negatively charged. Four, the nitrogenous bases point inwards, stacked like coins, perpendicular to the axis.
Held together how?
Hydrogen bonds between the bases on opposite strands, and here's the key insight. A purine base, the bigger ones, A or G, on one strand always pairs with a pyrimidine base, the smaller ones, T or C, on the other.
Which keeps the width constant.
Exactly, a consistent 2-nanometer width. And the pairing was specific. Adenine A always pairs with thymine T, forming 2 hydrogen bonds. Guanine G always pairs with cytosine C, forming 3 hydrogen bonds.
The Watson-Crick base pairs, and that explained Chargaff's rules perfectly. A matches T, G matches C.
Perfectly. And it meant the two strands were complementary. If you know the sequence of one strand, say AGG-TCA, you automatically know the sequence of the other, TCKQT.
The structure itself suggests function.
Absolutely. It immediately suggested how DNA could do its main jobs. First, store information. The sequence of bases is the code. Second, replicate itself for inheritance. Because of complementarity, you could unzip the helix, and each strand acts as a template to build a new partner strand. Pass on the information accurately.
Mind-blowing, though how the code was read, expression that came later.
That was the next chapter. But the structure was the key that unlocked everything else.
So the double helix is iconic, but the story of DNA's structure and function doesn't stop there, does it? There's more complexity.
Oh, definitely. For instance, in 1963, Jerome Vinograd discovered DNA supercoiling.
Supercoiling, like twisting a rubber band.
Exactly like that. He found circular DNA molecules could twist upon themselves, becoming much more compact, like a tangled old telephone cord.
Why does it do that? Is it just getting tangled?
No, it's actually regulated and vital, especially negative supercoiling, where the DNA is slightly underwound. This helps compact the incredibly long DNA molecules in our chromosomes so they fit inside the nucleus.
Okay, compaction makes sense. Anything else?
Yes, it also helps. That underwinding puts a strain on the helix, which actually makes it easier to separate the two strands locally.
Which you need for?
Replication, when you copy the DNA, and transcription when you read a gene to make RNA. So supercoiling influences DNA accessibility.
And there are enzymes that manage this twisting and untwisting.
Yes, topoisomerases. Amazing molecular machines. Type I enzymes make a temporary cut in one strand to relax supercoils. Type II enzymes cut both strands, pass another part of the DNA molecule through the break, and then seal it up again.
Wow. They can untie knots.
Literally. They can untangle knots and separate interlinked DNA circles. Absolutely essential.
And you mentioned these are drug targets.
They are. Human topoisomerase II, for example, is targeted by cancer drugs like etoposide. The drugs let the enzyme cut the DNA, but block the resealing step. This leads to DNA breaks, which preferentially kills rapidly dividing cells, like cancer cells.
Fascinating. Okay, what other properties of DNA are important? Melting.
Right. Denaturation or melting. If you heat DNA, the hydrogen bonds holding the strands together break, and the double helix unwinds into 2 single strands.
And the temperature that happens depends on the sequence.
Yes, the melting temperature or DNA rich in GC base pairs has a higher TAME because GC pairs have 3 hydrogen bonds compared to AT's 2, so it takes more heat to separate them.
But the really cool part is they can zip back up again.
That's renaturation or reannealing, discovered by Marmer and Doty in 1960. If you cool the denatured single strands slowly, the complementary sequences can find each other again in the complex mixture and reform stable double helices.
It's like they know their partner.
In a way, yes. This ability to hybridize specific sequences is the foundation for so much of modern molecular biology, DNA sequencing, PCR, gene probes.
And studying how fast DNA reanneals told us something about genome organization.
A lot. When you shear DNA into fragments, denature it, and then measure how quickly it reanneals, you get different patterns.
How so?
For simple genomes like viruses or bacteria, most sequences are unique or present in few copies. They reanneal relatively slowly in a smooth curve.
But complex genomes, like ours.
Plant and animal DNA reannealing curves show distinct phases: a very fast phase, a medium phase, and a slow phase.
What do those phases represent?
Different classes of sequences based on how repetitive they are. The fast reannealing fraction is highly repeated DNA. Short sequences, sometimes just 5 or 10 base pairs, repeated maybe millions of times.
Like satellite DNA.
Exactly, often found near centromeres. This class also includes things like mini-satellites and microsatellites, those short tandem repeats, STRs, that vary between individuals.
That's the basis of DNA fingerprinting, right?
That's it. The number of repeats at specific STR loci is highly variable, creating unique profiles used in forensics and paternity testing.
Okay, so that's the highly repetitive stuff. What's the medium fraction?
The moderately repeated fraction. Sequences repeated maybe hundreds to tens of thousands of times. Some code for essential molecules like ribosomal RNA or histones, where you need many copies. But most are actually non-coding sequences scattered throughout the genome.
Like signs and lines.
Precisely. Short interspersed elements like the ALU sequence in humans, and long interspersed elements like L1. They're remnants of transposable elements, which we'll get to.
And the slowest fraction?
That's the non-repeated fraction. These sequences are present in only one or a few copies per genome. This fraction contains most of the protein-coding genes, your typical gene.
So a genome isn't just genes. It's this complex mix of unique sequences and vast amounts of repetitive stuff.
A very complex landscape.
Yeah.
And it's not static either. Genomes evolve. They change.
How do they change? Duplication seems like a big way.
A huge way. Sometimes the entire genome duplicates polyploidization.
The whole set of chromosomes gets doubled.
Or even tripled or quadrupled. It's surprisingly common in plants. Think wheat, cotton, potatoes, bananas. It provides a massive amount of genetic redundancy.
Extra copies of every gene.
Right, which frees up one copy to potentially mutate and evolve a new function without losing the original function. It's a major engine for evolutionary innovation.
And there's a theory this happened in our own ancestry.
Yes, the 2R hypothesis. 2 rounds of whole genome duplication early in vertebrate evolution. Comparing our genome to simpler chordates like Amphioxus seems to support this idea. It might explain some of our vertebrate complexity.
But whole genome duplication is rare in animals, right? More common is just duplicating single genes.
Much more common. Gene duplication happens frequently, often through errors during meiosis, like unequal crossing over. A small segment of a chromosome gets copied.
What happens to these extra gene copies?
Most often they accumulate mutations and become non-functional relics called pseudogenes. We have lots of those.
Like in the globin gene family.
Exactly. We have genes for embryonic, fetal, and adult hemoglobin, all evolved from duplication of an ancestral globin gene. Some related copies are now pseudogenes. But sometimes...
Sometimes the duplicate evolves a new, useful function.
Exactly. Or it might evolve to be expressed at a different time or in a different tissue. This is how multi-gene families arise, providing related but specialized proteins.
So duplication provides raw material. What about genes moving around, the jumping genes?
Barbara McClintock's discovery, transposable elements. She proposed this back in the 1940s from her work on maize kernel colors. She saw patterns of variation that suggested genetic elements were physically moving, causing unstable mutations.
And people didn't believe her at first.
Not really. The prevailing view was that genes were stable, fixed points on chromosomes. Her ideas were too radical then, but she was absolutely right. She eventually got the Nobel Prize for it.
So how do these elements jump?
There are 2 main ways. DNA transposons use a cut-and-paste mechanism. An enzyme called transposase cuts the element out of one location and inserts it somewhere else.
Okay. And the other way?
Retrotransposons. They use a copy and paste mechanism. The transposon DNA is first transcribed into RNA. Then, an enzyme called reverse transcriptase makes a DNA copy of that RNA, and that DNA copy gets inserted into a new location in the genome.
Like retroviruses work.
Very similar mechanism. And these retrotransposons are incredibly abundant in our genome. Those signs and lines we mentioned, like ALU and L1, they're retrotransposons. Over 40% of the human genome is derived from them.
40%? That's astonishing. What do they do besides copy themselves?
Their impact is huge and varied. They can disrupt genes if they land inside them. They can carry bits of adjacent host DNA with them, shuffling genomic regions. They can contain regulatory sequences that alter the expression of nearby genes. Sometimes parts of them even get incorporated into host genes, creating new protein variations.
So they're major drivers of genome evolution and variation.
Absolutely. And there's even recent evidence suggesting, for example, that L1 retrotransposon activity might be higher in brain cells, potentially contributing to differences between individual neurons. Still an active area of research.
Okay, so we have this complex dynamic genome. How did we actually figure out the entire sequence? That seems like a monumental task.
Monumental is right. But technology advanced rapidly. The first bacterial genome was sequenced in 1995. Yeast, the first eukaryote, in 1996.
And then the big one, the Human Genome Project.
Yes, a huge international effort. A rough draft was announced in 2001 and a more finished version by 2004, sequencing all 3.2 billion base pairs of our DNA.
And weren't there some big surprises, like the number of genes?
That was perhaps the biggest shock. Pre-project estimates were often 50,000, maybe even 100,000 or 150,000 human protein coding genes.
And the actual number?
Turned out to be much lower. Current estimates are around 20,000 to 21,000 protein coding genes.
That's not much more than a simple worm like C. elegans.
Pretty much the same ballpark. It really drove home the point that organismal complexity isn't just about the number of genes.
So what else contributes to complexity if not just gene count?
Things like alternative splicing. A single gene in humans can often be spliced in different ways after it's transcribed into RNA, producing multiple different protein variants, isoforms, from that one gene. Vertebrate genes tend to have more complex structures with more introns, allowing for more alternative splicing possibilities compared to, say, worm genes.
So we get more protein diversity out of fewer genes.
Exactly. And complexity also comes from the intricate networks of gene regulation, how genes are turned on and off at the right time and place. Much of that is controlled by sequences in the non-coding DNA.
Which brings us to comparative genomics, comparing genomes to find important bits.
Right. The logic is, if a sequence has been conserved across millions of years of evolution between, say, humans and mice, it's likely doing something important.
Even if it doesn't code for protein.
Especially if it doesn't code for protein. Remember, only about 1.5% of our genome codes for protein. When we compare human and mouse genomes, we find about 5% of the sequences are highly conserved.
So that extra conserved stuff, the non-coding bits,
Yeah.
they must be important.
That's the strong inference. They likely contain regulatory elements, sequences that structure chromosomes, perhaps genes for functional RNAs we haven't fully identified yet. Projects like ENCODE are trying to map all these functional elements.
Okay, comparing humans and mice is one thing. What about comparing us to our closest relatives, chimpanzees? What makes us uniquely human genetically?
Well, we share a common ancestor with chimps, maybe 5 to 7 million years ago. Our genomes are incredibly similar, sometimes cited as 98–99% identical at the single nucleotide level. But that remaining 1–2% difference actually amounts to millions of single base changes, plus differences in gene duplications, deletions, and rearrangements. There's about a 4% difference if you include insertions and deletions.
Are there specific genes that seem to have changed rapidly in the human lineage?
Yes. Researchers look for genes showing signs of accelerated evolution in humans compared to chimps. One famous example is FOXP2, a transcription factor involved in brain development and speech. The human version has specific changes.
A potential language gene.
It's likely more complex than that, but it's definitely implicated. And interestingly, when they sequenced the Neanderthal genome,
Our extinct cousin.
right, they found Neanderthals had the same version of the FOXP2 gene as modern humans, which hints they might have had sophisticated vocal communication too.
And we know there was interbreeding.
Yes, the Neanderthal genome sequence showed that modern humans of European and Asian descent have about 1–4% Neanderthal DNA in their genomes. Some of those Neanderthal gene variants might have even been beneficial, perhaps related to the immune system.
So we're carrying a little bit of Neanderthal history within us. Fascinating. Finally, what about variation within the human species? We're 99.9% similar, you said?
At the single nucleotide level, yes. Any two people differ by about 3 million single nucleotide polymorphisms, or SNPs, on average. But we're also finding a surprising number of rare variations, possibly because our population has grown so rapidly recently.
And it's not just SNPs, there are bigger differences too.
Yes, structural variants are also hugely important for human variation and disease. These include deletions, duplications, insertions of sequences, and inversions where a segment of a chromosome gets flipped.
Copy number variations, see in these?
Exactly. Variations in the number of copies of specific genes or DNA segments. These CNVs have been linked to susceptibility to various conditions, including autism, schizophrenia, and some developmental disorders.
How do researchers find these associations between variants and diseases?
A major tool is the Genome-Wide Association Study, or GWS. You take thousands of people with a particular disease and thousands without, and you scan their genomes for SNPs or other variants that are statistically more common in the affected group.
Like the CFH gene and macular
That
degeneration.
was an early success story for GWAS, yes. It identified a strong association. GWAS has found hundreds of variants linked to common diseases, but often each variant only explains a tiny fraction of the risk. There's still a lot of missing heritability.
What helps in these studies? Haplotypes.
Haplotypes are really useful. Because recombination isn't perfectly uniform across chromosomes, SNPs located close together tend to be inherited together as a block, a haplotype.
So you don't need to check every single SNP.
Exactly. You can check representative tag SNPs for each haplotype block. The International HapMap project mapped these common haplotypes across different human populations.
Giving insights into population history, too.
Yes. For example, the specific long haplotype associated with lactose tolerance in Europeans is found at high frequency in populations with a long history of dairy farming. It's a classic example of strong positive selection in recent human history.
And sequencing whole genomes is getting cheaper all the time.
Dramatically cheaper.
Yeah.
From billions for the first human genome project down to around $1,000 or even less now for a whole genome sequence by 2014 and continuing to drop. This is opening the door to personalized genomics in medicine.
Wow. Okay, so from Mendel's P's through discovering DNA, mapping genes, sequencing genomes, understanding variation, it's been an incredible journey.
It really has. Yeah. And I think the big takeaway is just how dynamic the genome is. It's not the static library we once imagined. It's constantly being reshaped by duplication, transposition, recombination, selection.
That dynamism creates the diversity of life and also the differences between us as individuals.
Exactly. It fuels evolution, adaptation, and unfortunately, sometimes disease as well.
It's really clear we've learned so much, but also that there's still so much we don't understand, especially about all that non-coding DNA, the dark matter of the genome.
Absolutely. And how all these parts, coding and non-coding, interact in complex regulatory networks. That's where a lot of the focus is now.
So thinking about the future, as genome sequencing gets even cheaper and more widespread, what ethical or social challenges do you foresee becoming more prominent as this very personal information becomes easier to access?
That's a huge question. Issues around genetic privacy, potential discrimination based on genetic predispositions, equitable access to genomic medicine, how we interpret probabilistic risk information. These are all things society is grappling with and will need to continue addressing very carefully.
Definitely food for thought. Well, thank you for guiding us through this incredible deep dive into the nature of the gene and the genome. It was really comprehensive.
My pleasure. It's a fascinating story and it's still unfolding.
We hope you listening found it compelling too. Thanks for joining us.
Until next time, keep exploring the amazing biological world around you and the equally amazing one inside you.